I'm Walter Smith, CEO of Confluence Life Sciences, a drug discovery and development company based in St. Louis. At Confluence, we focus on kinases, enzymes that are important in cancer, autoimmune disease, and chronic inflammatory disease. At Confluence, we've developed a proprietary technology platform we call Connect. It allows us to discover inhibitors of kinases in a fraction of the time it takes traditional pharma approaches. Part of the Connect technology platform is a novel thiol-directed chemical library from which we can discover and develop potent and selective inhibitors of kinases. We've partnered with Harness, Dickey, and Pierce, the Milwaukee School of Engineering Center for Biomolecular Modeling and 3D Molecular Designs to create a physical model of one of our targets, TAK1. TAK1 is important in pancreatic cancer and other hard-to-treat cancers. Today, Jim Blinn is going to describe this physical model and how compounds interact with the active site.
Hello, I'm Jim Blinn. I'm the head of computational chemistry at Confluence. And before you see a 3D printed model of an enzyme that we are currently working on called TAK1. It is a kinase and has a typical kinase fold. In the kinase fold, there is an end lobe, which you can see over here, which is primarily composed of beta sheet, although it also has one prominent helix called the C helix. It has a C lobe, which is primarily composed of alpha helix, although we have deleted most of the alpha helices from the model for clarity. In between the two lobes is the active site cleft in here. And connecting the two lobes is what's called the crossover or hinge region, which is here colored in green, which is responsible for making some characteristic hydrogen bonds to ATP. Also, highly conserved in part of the catalytic mechanism, you have an aspartate, and the coloring here is that oxygens are red, nitrogens are blue, and sulfurs are yellow. So you have the aspartate, you have a lysine, and you have a glutamate coming off the C helix. Also in this model, there is a water right here, colored red, which is present in the crystal structures that have been published to date, and which turns out to be important for our purposes. Now, although there is a high degree of conservation in the kinome of the catalytic residues and the interactions they make, because all of these kinases have to bind ATP, there are points of difference among them. And one of the notable differences which we focus on at Confluence is location of sulfurs in the active site. There are over 500 kinases in the kinome, in the human kinome, but there are only a little over 200 that have a cysteine with a reactive thiol in the active site, and in those 200, the cysteine can be found in any of a number of locations. Here, right before the catalytic asp, is certainly one of them, but they can be found on the glycine-rich loop here. They can be found on the hinge. They can be found on the outside of the active site very prominently in a number of well-known kinases. The role of a kinase is to bind ATP, which then is able to donate a phosphate to another kinase, typically. In this case, serine or threonine on another kinase. And this is part of the signaling cascade that is important in things like pancreatic cancer and inflammation. And TAK1 in particular is involved with both of these. You can see here we have an adenosine from a crystal structure of TAK1, and the phosphate would be coming out approximately here. It makes 2 hydrogen bonds to the hinge region and makes additional hydrogen bonds to the protein, some of them mediated by water. However, you'll note that in this particular case, it really is not involved in hydrogen bonding to this water, nor is there any sort of an interaction of any importance with the cysteine that we have been focused on. Now, the reason we want to focus on cysteines is that while there are over 500 kinases, there are only a few, relative few, in this case approximately 40 kinases that have the cysteine in this particular location. Thus, our approach using the Connect platform is to interrogate the protein with various types of covalent inhibitors and deduce where we can make a covalent bond which will isolate the binding activity to just the class of kinases which has a cysteine, in this case, in this particular location. And there are only 40 or so kinases that have this particular cysteine. Thus, by doing this, we can cut down the specificity problem by an order of magnitude. Here we have a known covalent irreversible inhibitor of TAK1, which is called oxazianol. And this exemplifies the covalent approach, but is definitely not a molecule we would be interested in making or working with. It is very complex. It makes a single, rather weak hydrogen bond to the hinge. It is likely to have very poor properties. It would be difficult to synthesize analogs. It's a very complex molecule, but it's a good tool molecule. Nonetheless, where it is of interest is that it does make the covalent bond to this cysteine in TAK1. Now, we have been able to take this sort of information that we have gleaned from the use of the Connect library, and to deduce how to make some very potent and proprietary novel compounds that will bind to TAK1, some reversible, some irreversible, and this is the focus of this program at this point. So I'd like to thank you for listening to this talk, and I hope it'll allow you to examine the model with a little more knowledge. And I would like to thank the Milwaukee School of Engineering for their ability to provide this 3D printed model. And I'd like to thank Harness, Dickey, and Pierce for their assistance in patenting the novel compounds that we have been able to develop using this structure-based approach.